So thank you for spending your next 3 hours with me learning about gene curation. The way that I have this talk structured is I have maybe about an hour and 10 minutes or a little bit more worth of slides, and then I wanted to leave plenty of time for questions and discussion. And then, of course, we have an hour and a half of workshop, too. So unless you have a burning question, I would say maybe hold it until I'm going to talk about the framework. Then I'll take a break if anyone has any questions, and then I'm going to talk through some examples. So feel free to come up to the microphones during those times and ask some questions. Okay, so I'm going to talk to you about curating gene disease validity using the ClinGen framework today. I have no disclosures. And so the agenda, as I mentioned, is an overview of the gene curation process. Then we're going to do some selected examples, and then approximately 20 minutes of Q&A if anyone has questions. I'm not sure if people have shown this today, but this is the infamous ClinGen infographic. So ClinGen is pretty much interested in expert curation, and we have 3 different questions that we ask when we do expert curation within our disease-specific groups. And the one that we'll be talking about largely this afternoon is this gene associated with disease, which is clinical validity. And I wanted to specify a little bit what we mean about clinical validity when we're saying it in relation to gene curation, because many people will think of it as the clinical validity of a laboratory test. And in that case, it involves the test's ability to consistently and accurately detect or predict the outcome of interest. But for the clinical validity of a gene-disease pair, we're really asking the question, how strong is the evidence that a gene is related to disease? And this is important because essentially for the ACMG AMP sequence variant interpretation guidelines, so for variant curation, which we'll learn about tomorrow, it's essentially a given that any variant that you're curating is in a gene that is convincingly associated with disease. So gene curation essentially is the step above variant curation. So you need to prove that a gene is associated with disease before you can say anything about the variants in that gene and whether they're damaging and what type of phenotype or type of disease they would be causing. And it's also, clinical validity and gene curation is also important because in research, it is important to try and identify what evidence is necessary to solidify the role of a candidate gene in disease. So what types of experiments do you think you should be doing to solidify a gene-disease association? And we'll talk about that too. And I just wanted to point out the importance of gene curation with a specific example of the ClinGen expert panel for Brugada syndrome. And this was published in Circulation semi-recently in the last maybe year and a half. And the group had actually found when they pooled 21 genes from clinical testing panels that had been claimed to be associated with Brugada syndrome, of the 21 genes, 20 of them were actually refuted for the gene-disease association, and 1 gene, SCN5A, was associated with Brugada syndrome. So gene curation really is critical for interpretation of variants and for medical care and clinical care of patients. So ClinGen actually came up with a systematic method to evaluate clinical validity of gene-disease pairs. And we defined the criteria needed to assess gene-disease validity, experimental and genetic evidence, and I'll talk about that. It is a semi-quantitative framework, so we are using a point scoring system. And this actually allows users to more methodically classify the validity of a given gene-disease pair. However, it's semi-quantitative, so it's not intended to replace more rigorous statistical methods, such as those established for genome-wide association studies or for polygenic risk scores, multifactorial disease risk, et cetera. This is the paper. Hopefully that had been emailed out to all of you, and if it hadn't, it's in the workshop folder. And this is from American Journal of Human Genetics. I have the Pubmed ID listed there. And this details the framework, and I'll be spending most of this time also telling you about the framework. You can also find it on the ClinGen website at clinicalgenome.org, and this is just a screenshot here. If you go to gene disease validity and training modules, there's the publication, and then the standard operating procedure, the SOP, which there's also a copy of the most recent one in the gene curation workshop folder. So that will be there available for you. And if you look here, these are the buckets that we use to describe clinical validity terms. Definitive, strong, moderate, limited, no evidence reported, and then for conflicting, we have both disputed and refuted. And we'll talk a little bit about all of these. So for developing the framework, what we did is we chose gene-disease pairs across a range of different disease areas, 33 different pairs to be exact, and we actually had dual curation of these pairs. And after the two curators each scored the gene-disease pair, we had experts review those scores, and then we measured the concordance. And I should mention that for at least within the ClinGen framework, a gene curation is a gene, a disease, and a mode of inheritance. So if a gene is supposed to be associated with multiple diseases, or if a disease is even posed from multiple inheritance patterns, you would do separate curations for those. If you find that there's some sort of overlapping spectrum, it's something we call a lumping and splitting conundrum. Maybe you might need a semi-dominant gene curation instead, but usually it's one gene, one disease, one mode of inheritance. And so when we looked between the dual curators, concordance was actually 94%. So 29 out of the 31 genes were concordant across the curators, which really showed that this methodical, semi-quantitative gene curation framework was doing its job as we had hoped. And then there was pretty high concordance between curators and experts too, 88%. And the 4 genes that did differ were likely borderline genes. So those ones differed by only 1 level. And that's actually the importance of expert reviewers. When there are genes that are truly borderline, say, for example, between limited and moderate, or moderate and strong, it's really someone with a very strong knowledge of the disease area that can come in and say, in my experience level, I feel like this should be strong, or in my experience level, it should be limited. So we were pretty okay with how concordant this framework had proved to be. And so this is the workflow here. Because we want to be as transparent as possible and because all of the curations go on the website, what we largely curate actually is from the literature. And we have 2 specific scoring frameworks, genetic evidence and experimental evidence. So we'll curate both of those and then assess them and complete a summary matrix, which is essentially pulling the scores from genetic and experimental evidence and adding them together to correspond to one of the clinical validity buckets that I showed you on previous slides. And then we will assign a clinical validity classification. And then we'll bring that to most likely one of the clinical domain working groups like the Brugada syndrome group that I showed you in ClinGen that has a bunch of experts, national and international experts that will weigh in on that gene curation, change anything that they feel should be more concordant with the disease area, and then finalize the curation. And this is the summary matrix here. So you have the genetic evidence. You can score 0 to 12 points for genetic evidence, and this is both case level data and case control data and segregation data. And experimental evidence, which you can score up to 6 points, and these include things like biochemical function, animal models, a functional alteration in patient cells, et cetera, and I'll tell you all about that shortly. And this can total up to 18 points. And as you can see, here are the corresponding numbers for each bucket. So limited, for example, is 1 to 6. You'll notice that strong and definitive are actually the same amount of points, 12 to 18 points. However, the way that you bump up a gene curation from strong to definitive is that the gene curation has been replicated over time. And so we wanted to see that there are more than 2 publications with human data implicating the gene in disease over more than 3 years, just to give researchers enough time to potentially refute or dispute or publish conflicting information. And so in that case, even if you would have the same score, you would bump from strong to definitive. And I should mention, actually, there is a section here to list contradictory evidence. We'll talk about this in more detail, but contradictory evidence can be very variable, I should say. And we often need experts to weigh in on how serious this contradictory evidence is in a gene-disease relationship. And we'll talk more about that too. So to start with, we start with a literature search. And I'm sure you're familiar with many of these resources, but these are some of the resources that we really suggest to use. So online Mendelian inheritance in man, OMIM. Orphanet, which is more a rare orphan disease website. Gene Reviews, which is a book on the NCBI bookshelf that will talk about particular diseases. PubMed, of course, Google Scholar. I found that when you get started with a gene disease association, there can be two ways that you could get started. You could be doing, for example, exome or genome analysis. You don't necessarily have a given disease for that gene. You just have the gene and potentially a variant that had been identified in a person. So in that case, I found that it's very helpful to get a general sense of what has been published in the literature about that gene by looking at a general resource such as OMIM. Within ClinGen, most of the time we have disease-specific expert panels, and they have particular disease areas. So in that case, you might be getting a gene and a disease and a mode of inheritance already. So you could go straight to searching about that specific disease for that gene. But in general, if you're doing exome genome analysis, you really just want to search about the gene and get a general sense, because your literature search is going to differ if you have a limited gene or what seems to be a limited gene versus a very definitive gene. You might just be looking at reviews for very definitive genes, whereas for limited genes, you want to find pretty much every single publication of human evidence that you could score, because it could really make a difference. So it's really helpful to start by looking at a general resource and seeing, does OMIM even have a disease listed for this gene? Does it have multiple diseases listed? How similar are the diseases? What are the inheritance patterns of those diseases? I like to go to Genetics Home Reference, actually, because that's a really good reference for basically the lay population or patient population. And so a lot of times, if there's no gene disease association listed on Genetics Home Reference, there isn't a huge amount of information. Whereas if there's a very nice, thorough Genetics Home Reference entry on the gene disease association, for example, more than 30 variants have been seen in patients with this disease, you know that you're looking more at a strong definitive type gene disease association. So for Pubmed searches, we often suggest to start broad and inclusive for your initial search. Really just as a first pass, maybe just searching the gene symbol. That being said, you want to check HGNC to see if there are old symbols or aliases, because a lot of times, particularly experimental evidence was published under an old gene name before they changed the gene name to whatever you have right now. And so you'll miss some of that experimental evidence sometimes if you don't know the old aliases for the gene name. So take a look and make sure there aren't any multiple names that you should be searching. So if your broad initial search comes out with a ton of results, like 122 results in this case, you want to narrow it down a little bit. So then what we suggest is to just do the gene and the disease. In this case, if you have a disease or if you know what type of disease you're looking for. And so in this case, it would narrow it down from 122 results to 7, making it much more manageable. You could also do other searches to find relevant information. One that is one of my favorites is I try to go out of my way to look for animal models, because those are quite valuable to experts and high scoring for experimental evidence. So you can search for gene and animal, gene and function if you want to try to find a little bit more experimental evidence. OMIM is actually pretty helpful in the gene function section or the biochemical features sections. Gene Reviews also has a molecular genetics section. So just looking through all these different sections in the resources that I had mentioned previously will give you some more results potentially to curate. And then another big thing that we actually find is references in other papers. So a lot of times in the discussion of a paper that identifies human variants for a particular gene that's a gene discovery paper, they'll cite the mouse model. And so then you would have a reference for an animal model that was just given in a discussion. Although we do suggest that you always, unless it's a really definitive gene disease association, which I'll talk about, that you should always curate the primary literature. So if you find a reference in a discussion or a reference to another experiment, you should go to that paper and curate that paper directly. So you should also evaluate your search results, because maybe you are getting a ton of search results. And ask yourself, what should you be curating? As I mentioned, we really do suggest curating the primary literature. At least for ClinGen, we put all of our curations on the internet and make them public to everyone. So we feel that curating the primary literature where something was identified is the most useful, instead of curating for example, a genetics home reference or a gene reviews article or something like that. As I said, not all search results will be relevant. You should curate all genetic evidence until the maximum score is achieved, or at least you reach the highest classification you would reach. So we'll have a little bit of a discussion about curating definitive gene-disease relationships, but there are different schools of thought. Some people feel that once you reach the definitive scoring, you don't need to max out genetic and experimental evidence. And we have little summaries of our gene curations, kind of like the evidence summaries that you have for ClinVar and variants, that kind of can give you a sense that gene, for example, BRCA, that this gene has been, variants in this gene have been found in hundreds of patients, things like that, that can give you a sense that a gene is very definitive without you having to spend all of this time maxing out genetic and experimental evidence. But I would say at least curate until you could get to the highest level that you can for gene-disease association, so at least to definitive, not necessarily maxing out the categories. You should also curate across the breadth of experimental evidence available until at least getting to the highest you can, so potentially definitive. Review articles are very good for definitive gene-disease associations, so gene disease pairs with abundant information. You might want to search specifically for gene and disease and review. And when you have a sufficient detail, and we'll talk about this in the workshop, to curate a review article, you may be able to curate that if it's a very definitive gene-disease association, especially if it has hundreds of patients in it or a table with variants that you could very easily score and get to definitive. In that case, it would be acceptable to curate the review. But you do have to have enough information. You can't just have them saying 50, 60 plus people have variants in this gene. You do need to see the variants and make sure they're convincing to you. And to look for replication over time, you need to find the original paper with the proposed relationship in humans. OMIM is pretty useful for that in the allelic variant section. Gene reviews, you can also look in the molecular genetics section, pathogenic allelic variants. But extract information from the original publication just to make sure it is the first publication, especially if you're looking at replication over time. And for very definitive genes, you should anyways use a recent review article to rule out any contradictory evidence. So maybe there's an overwhelming amount of evidence, but recently, for example, with population databases or something, much of this evidence was overturned, something like that. So you do want to just try to find a very recent review article and take a look at it and make sure that the gene is definitive as you thought. So this is the genetic evidence scoring matrix, and we'll go into this in detail. Kind of a broad overview is, it is separated into case level data and case control data. So case level data will include scoring variants and the variant evidence and segregation. And case control data will score two types of case control studies, single variant analysis and aggregate variant analysis. The total allowable points for genetic evidence is 12 points. And each type of variant, because all the cases are separated by types of variants, has a maximum total score that you can score using those types of variants. For example, a proband with another type of variant that's not null, so most likely a missense variant, you can only get a max of 7 points with those types of probands. And we'll talk about this in more detail too. So for scoring case level data, each case can be given points for the variant evidence, and this is split, if I go back, this is split by mode of inheritance actually, so you're gonna do different scoring for autosomal dominant and X-linked than autosomal recessive, which is why I mentioned for a gene curation, it's a gene, disease, and a mode of inheritance, because there's specific scoring for each mode of inheritance in the genetic evidence matrix. So you can score for the variant evidence in the context of the appropriate mode of inheritance, and also segregation evidence if applicable, and we do have some restrictions on the size of families that you would use for that. And so this is a zoomed in version of the case level data. So we have it split out for autosomal dominant X-linked versus autosomal recessive. De novo variants receiving the highest amount of points. We have a suggested default point range for each type of variant, and I should say it's a case. So And this is why inheritance is important. For autosomal recessive, you have to have 2 variants, and they have to be convincingly proven to you that they're in trans. So you're scoring a case. You're not necessarily scoring a variant. You're scoring a person. So the highest amount of points for autosomal dominant or X-linked is a de novo variant. And for the default, we have 2 points. However, we do give a scoring range for each type of variant. We do suggest that people start out by scoring with default scoring. And as you get a sense more of the nuances of gene curation, you can start to upgrade or downgrade. And in the standard operating procedure, we do provide some specific examples of when you should downgrade and upgrade. And we'll talk about some too in the workshop. And then the one that scores a little bit less than that is a proband with a predicted or proven null variant at a 1.5 per case. And then for missense variants or non-loss of function variants, you really need to be convinced that there's at least some impact to the gene function for the case to count. So if it's just a missense variant that really doesn't have any experimental evidence or computational predictors predict that it's going to be benign, or if it's high frequency in the general population, you wouldn't want to score it because you're not convinced that this could ever be disease-causing in that case. For autosomal recessive, as I mentioned, you do have to be convinced that the variants are in trans and not in cis. So if that means the family has been sequenced and you find 1 variant in each parent, that is convincing evidence that they're in trans. If they mention something else about the phase, you want to be sure that they're not on the same allele. So for loss of function or predicted proven null, those are higher scoring, so those are 2 points. And for non-loss of function variants, most likely missense, maybe in-frame deletions, things like that, you would usually give 1 point. You could consider assigning more points if the gene is known to be fully constrained for loss of function variants, and I'll show in Nomad the constraint scores and where you would find that. Fewer points if there's alternative splicing, or fewer points if a loss of function variant is at the end of the protein, essentially, and nonsense-mediated decay is not predicted to occur. And also, you want to consider the statistical expectation of de novo variation in the gene in question. Is it a gigantic gene that you would just be having random de novo missense variants in the gene, like Titin or something? So questions to consider when scoring a variant, and Stephen will talk about this in major depth tomorrow when he talks about variant curation, but this is essentially, I like to think of it, rapid variant curation for an entire gene. And in theory, you can't do a true variant curation before you do a gene curation, but you need to just be convinced that a variant is suspicious in this case. So usually what we'll do is we'll go and look to see if this variant is present in population controls, essentially, in this point. And if so, what frequency? So we go and look at it in Nomad and see if it's high frequency. You want to consider the prevalence of your disease and the penetrance of your disease. Is your disease a later onset condition that there could be people in Nomad that would have this condition? Or is it very early onset, high penetrance? If the variant is a missense variant, you want to look at some computational predictors. Do they predict that the change is damaging? You might also want to look at conservation. Is this residue very highly conserved? Or do multiple other species have this specific variant change too? If they do, it might be less evidence that the variant is suspicious. And most importantly, does the paper contain functional evidence to support that the variant has a damaging effect on the gene? If so, you would be much more suspicious of particularly of missense variants. So we also have segregation scoring. And this is just a little bit of an overview of segregation scoring. So we actually have a quick and dirty, and I'll show you the calculation that we use, way to calculate LOD scores through the ClinGen framework. I should mention, if a LOD score is published by the authors, you should definitely use that LOD score because they're using programs to calculate the LOD scores, and it's a lot better than our rapid estimation of a LOD score. But essentially, we will calculate a quick and dirty LOD score. We'll sum them across all of the families. We'll consider what types of sequencing were done. So if it was candidate gene sequencing and they really haven't looked at any other gene at all in this patient, you would receive less score for the segregation in that. It's less convincing. Whereas if a person has been fully investigated, so either an exome or genome, or if they did linkage analysis and sequenced all the genes in the linkage region, you can be a little bit more convinced that the segregations are meaningful. So we can give a little bit more points in that case. The total number of segregation points that you can award are 3 points at this point. So these are the LOD score calculations that you would use. So for dominant disease, you're going to do 1 over 0.5 to the number of segregations. For recessive disease, we factor in both affecteds and unaffecteds, and this is the formula that we use here. For unaffected individuals, they have to have the same chance of receiving the damaging variant as the unaffected, or as the affected individuals, and I'll show you, it's usually siblings or someone in the same generation. Like you wouldn't want to do parents that are above or something like that, but I'll show you some examples. So we have a size restriction for counting segregations. For dominant gene disease associations, you have to have at least 4 segregations in a family to count for this LOD score calculation. For recessive, you can calculate a LOD score in a family with greater than or equal to 3 affected individuals. And to be very careful, unless your disease is highly penetrant and very early onset, non-segregations are phenotype positive, genotype negative. So affected individuals who do not have the variant. So this is an example. Many people will consider this a non-segregation, that the person has the variant, but they're not affected. However, this could be due to age-related penetrance, particularly in some conditions that we've curated recently, such as cardiomyopathies, that the penetrance is incomplete, or maybe something would show up later in life. And especially if they don't give you the age of the individual and the pedigree in question, we wouldn't necessarily count this as contradictory evidence or a non-segregation. However, if you have an affected individual here that does not have the variant, that would be counted and considered a non-segregation. Phenotype positive, genotype negative. So this is just an example of counting segregations, and this is also in the standard operating procedure, so you can refer to it at any time. But you can see the different colored lines counting towards all of the 4 segregations. This is actually including the mother here, who's an obligate carrier, who we're assuming is genotype positive because there are 2 genotype positive affected individuals on either side of her, so we can count her too. For disorders of reduced penetrance, it's safest to only use affected genotype positive individuals, except like this mother who, individuals who must be carriers because they have a genotype positive parent and a genotype positive child. So regardless of her phenotype, we could count her because she has the 2 individuals on each side. So in this case, because we have 4 segregations, if you go back to the formula here, we would put in 4 segregations here. And so when you do that, you get 1.2. But if you look back to the framework and go to a LOD score of 1.2, it's actually too low to even score. So you would have to have more families together to add up to a higher LOD score to even score segregation in that case. And I know this is a little bit confusing, but really with practice, it becomes more intuitive. So for autosomal recessive, counting segregations, as I mentioned, you count affecteds and unaffecteds. So for example, in pedigree A, the unaffecteds that you would consider are those that have the same risk of inheriting the variant and potentially being affected as the affecteds. So you wouldn't count the parents as unaffecteds. In this case, you would count the sibs. So in this case, we have 3 affecteds and 2 unaffecteds. And so you would put that in the formula that had affecteds and unaffecteds in it to calculate the LOD score. This one has 3 affecteds and 1 unaffected in this generation, and then 3 affecteds and 2 unaffecteds in this case. And so if you calculate all the LOD scores across these pedigrees, you get 1.5 for the first one using that equation that we had, 1.3 for the second one, 1.5 again, which is a total LOD score because they're additive of 4.3. And depending on how these individuals were sequenced, because I told you candidate gene analysis versus exome genome or all the genes in a linkage region being sequenced, they would score in between 1 to 2 segregation points with this LOD score. So now I want to talk to you about scoring case control data. As I mentioned, case control studies are classified in 2 ways. There's a single variant analysis and aggregate variant analysis. So in a single variant analysis, you're looking at enrichment of a single variant in cases versus controls. More than one variant can be analyzed in this case, but they should be independently assessed and have correction for multiple testing in that case. Whereas for aggregate variant analysis, maybe you're just looking at statistical enrichment of 2 or more variants or all the variants in a gene, your particular variants of interest in cases compared to controls. And you should assess each case control study independently to evaluate the quality of the study design. And this is why we left the framework so flexible, so we really do recommend consulting experts in this case, because it's so flexible that a case control study can score anywhere from 0 to 6 points. So literally, if you had 2 very well done case control studies, you could max out your genetic evidence. So it's very important to talk to experts in this case. And so these are the bullet points that we consider when evaluating the quality of a case control study: the variant detection methodology, power, statistical significance, and bias and confounding. And I'll talk about those a little bit in detail now. So for variant detection methodology, you really want to ask how the cases and controls were tested. Were they both tested in the same way? Were they sequenced for the entire gene under analysis? Or they genotyped for that particular variant? In this case, you want to pay attention to the methods section. And here's an example. Cases were sequenced for all exons in PALB2, and they identified a nonsense variant, and then they genotyped it in controls. So in this case, and this is a common way that they do things, in this case, cases and controls are not matched for variant detection methodology. The next one that you want to consider is power. And you're actually not expected to perform any power calculations. That would be too complicated. You really just want to consider the size of the study in relation to the prevalence of your disease. So you want to think about, did they analyze a sufficient number of cases and controls? And if you're unsure, you can ask experts in the particular disease area. You also want to consider if this cohort might overlap with potential other studies, and try not to score them both, because that would be double counting cases across. Or if you have a meta-analysis, you don't want to score all of the case control studies within the meta-analysis independently and then score the meta-analysis, because that would be double counting. The next thing you want to consider is bias and confounding. So how well matched are the cases and the controls? Are they matched based on ethnicity, location of recruitment, genetic ancestry, age, gender? Are they equivalently phenotypically evaluated? Have family histories been taken for cases and controls? And most of the time, it's not yes for all of these questions, but that's something you consider when you are awarding points, anywhere from 0 to 6. Was previous testing conducted on cases, maybe the most common cause of disease, hopefully ruled out in these cases? Is there an ascertainment bias because the cases came from families with a history of disease? And then you want to look at statistical significance. So essentially, I know many studies that publish genetic evidence will say that we sequenced this many cases and we found this variant and it was absent from controls, and that's where they'll stop. If there's no statistical comparison of enrichment in cases versus controls, that has to be scored within case level evidence as the affected individual. You need to have a statistical comparison between the cases and the controls to score in the case control section. And some examples of this are odds ratios. Maybe they're doing a Fisher's exact test with a p-value. Just some evidence that there's a statistical comparison. Relative risk, standardized risk ratio, standardized incidence ratio, hazard ratio. And in the cases when you're scoring them, you do want to make sure that there's a statistically significant difference in cases versus controls of enrichment. And it does depend on your disease area, potentially. I know that some experts, for example, in the cancer gene curation working group, will not score a case control study unless the odds ratio is 2 or greater. So that might be disease specific that you would set a cutoff like that. And so I want to give you an example from the breast cancer ClinGen clinical domain working group. And this was for ATM and breast cancer. It was an aggregate variant analysis of almost 30,000 Caucasian breast cancer cases referred to panel testing to Ambry. And they were sequenced by NGS for 16 susceptibility genes, and they were compared to a similar number of exact non-Finnish Europeans, the controls, and this excludes the cancer genomes within exact. And the variants were pathogenic or likely pathogenic in cases, and the control variants that were identified also deemed likely path and path by Ambry systems, so they were kind of just looking at pathogenic and likely pathogenic variants. Control path and likely pathogenic variants also had all of these different loss of function variants that are suspected to be pathogenic. And so you want to look at variant detection methodology. ATM was sequenced in cases and controls, so that was matched. How was the power? The total number of cases and controls, it was about 30,000 for the one and 27,000 for the other, so that's pretty adequately powered there. Did they evaluate the statistical significance and what did they use? So there was an odds ratio calculated and it was above 2, which was their cutoff for cancer group. And the p-value was very statistically significant. The confidence interval didn't cross 1. And you also want to consider bias and confounding. Controls were matched to cases based on ethnicity, but not genetic ancestry, location of recruitment, or age. And so they ended up scoring this 4 points out of a total 6 points. And it was due to the power of the study and the use of ethnically matched controls being some of that, and also the statistical significance. So closer to the high end, but I think I've talked to the groups that score a lot of case control studies, and they maybe have only had 1 or 2 that have been perfectly scored at 6 points. So this is pretty standard, I would say. So now we're going to talk about experimental evidence and scoring that. And the matrix looks kind of similar to the genetic evidence matrix here, and that you have a score range for each type of experiment, which we'll talk about, and recommended number of points per piece of evidence and a maximum number or maximum score. And the maximum score for all experimental evidence is 6 points. I should point out that the maximum score for each type of experimental category is maybe more important than it is in the genetic evidence section, because you can essentially stop looking for that type of experiment once you've maxed out because you can't score it anymore. For example, expression studies, you can find a ton of expression studies that the gene is expressed in your tissue of interest that is relevant to your disease, but you can only score 2 points total. So that's only a couple expression experiments. So at that point, you can stop looking for expression studies because you've maxed out using your expression studies. And we did this on purpose too, because we felt like an overwhelming amount of evidence of one type of experiment isn't necessarily super informative. You want to have animal models, you want to have expression, you want to have biochemical data in theory. And so all of the categories that we chose are consistent actually with the MacArthur et al. 2014 paper that evaluated what types of experiments should be used to classify variants. And so in the function category, we start off with biochemical function. And so the first one being if A and B are 2 genes that are similar, and their functions are similar, and they're involved in the same disease, you can score that as evidence. But one of these has to already be definitively associated with the disease. So a good example is potentially the RASopathy genes, that in the MAP kinase signaling pathway, many of them are very definitively associated with disease. So if you came along and had another MAP kinase or another kinase found in the same signaling pathway and said, it's downstream, it's upstream, it's very similar in function of this one that's definitively associated with a RASopathy, you could score that biochemical points. Another way to score biochemical function points is if the function of a gene is consistent with a patient phenotype. So many times this is for inborn errors of metabolism, or if you're curating for an enzyme, essentially, and you know the enzyme function and the product of the enzyme is lacking in the patient, something like that. For interactions, A and B need to be involved in the same disease area, and they could physically interact, they could genetically interact, and just like above, one of these has to be definitively associated with disease already. It can't just be a screen that's found multiple genes and none of them are associated with disease. For expression, and this is a bread and butter type experiment, but as I mentioned, that the gene is expressed in a tissue that's relevant to the disease, and you can score that. Depending on your disease area, you might need to score this in an animal model. For example, we do a lot of curation for hearing loss, and you really just don't get a lot of human cochlea because it's post-mortem tissue. And so we mostly have to score this in mice or rats or potentially zebrafish. So it does depend on your area of interest, your disease area. And potentially if expression is altered in a patient. Other experimental evidence categories, so functional alteration, which is really a bread and butter experimental evidence category. This can be in patient cells or non-patient cells. And this generally is the typical knockdown experiments or transfecting a variant into a cell line and showing that the function or the loss of the gene is causing something that is similar to the disease or as close as you can get to the function of the gene in the disease in either patient cells or non-patient cells. For model systems and rescue, that's a little bit self-explanatory, but essentially if you knock out the gene product and the phenotype is similar in an animal model to that in humans, or even if you have a human system and you can rescue, which is the second one that you would score in model systems. So you can either rescue in an animal model and add the gene back and it will rescue the phenotype, or even enzyme replacement therapy. If you add the enzyme back in a human and it causes rescue of the phenotype, you can score that too. And so as I mentioned, this is the summary matrix. So you take all your genetic evidence scoring points, your experimental evidence scoring points, total them, decide if it's been replicated over time, and come to a calculated classification. So for contradictory evidence, we considered giving this negative points to downgrade the score that you were giving the convincing evidence, but we found that the types of contradictory evidence were so differing and vast that it really was important to just document it and then consult with experts. So in the case where you find contradictory evidence, you would still use the spreadsheet that I'll show you and this framework to quantify or to organize all of your data, but you don't give it a final score. You just give it a contradictory evidence score, and then you consult with experts. And they decide whether you would end up scoring this as disputed or refuted, or that evidence is just too weak to overturn a gene-disease association. And you would just write a note of why the experts said this. I'll give you an example. And then score it as normal. And this leads on to how important expert reviewers are. And so at least in ClinGen, we said that experts can refine scoring in the matrices to better suit the complexities of a given disease area. So for example, setting an odds ratio cutoff. In hearing loss, and you'll see this example too, we end up with a lot of consanguinity. And so we will downgrade variants if they've been identified in a consanguineous family. Things like that. The only thing that experts really can't change are the maximum amount of points that you can give for a specific type of variant, or the maximum amount of points for genetic and experimental evidence. And you should really, once you specify this for a particular disease area, consistently apply it across all of the genes in that disease area. For example, that's what we did in hearing loss. We specified it in the beginning and then applied it across all 164 genes we did. And this is just an example of, it's a little small, of all of the gene curation groups that we have right now. Cardiovascular has a bunch of groups within it. We have the hearing loss group, hereditary cancer, inborn errors of metabolism, neurodevelopmental disorders, RASopathy, and even more at this point, brain malformations, mitochondrial disease, et cetera. So in conclusion, ClinGen has developed a gene curation framework, and they assemble groups of experts to apply this framework in particular disease areas. And standardized and transparent curation processes will help to improve variant interpretation and patient care in this case. All these gene curation training resources will be in your folder for the workshop, and they can also be found on the ClinGen website, very easily accessible. And so I'll stop at that point before I talk about some examples to see if anyone has any general questions on the ClinGen framework before we talk about examples. Okay, if not, we'll go on to the examples, and maybe applying it will be a little bit more informative, and if anyone has questions after that, we can talk after that. So I wanted to try and give you a limited, a moderate, and a definitive curation to give you an example of the types of evidence that you would see and the amounts of evidence in all of these curations. So the first one that we'll start with is PNPT1 and autosomal recessive hearing loss. This was curated and approved by the ClinGen Genetic Hearing Loss Clinical Domain Working Group. So as I mentioned, we like to get an overall sense of the gene and the disease area before we start a curation. So PNPT1 is a polyribonucleotide nucleotidyl transferase. It has two alternative names, and it's a subunit of the exosome complex. This is really just taken directly from the OMIM entry. We also like to get a sense of the variant spectrum. So in this case, this is not an open source, at least the professional version. The academic version is the Human Gene Mutation Database. We'll take a look at the variant spectrum that's posed in there for the particular gene. You could also just go to ClinVar and see what types of submissions have been submitted for this particular gene, just to get a sense of the variant spectrum, kind of giving you a sense of the mechanism of disease. Are there largely missense variants? Are there largely loss of function variants? Is there a mix? And how many are there? So in this case, in HGMD, there are 3 missense variants. There's 1 splicing variant. And we also like to see what diseases are posed to be associated with these variants, just to see how many diseases we might be working with. So multiple diseases, severe delays, seizures, microcephaly, sensorineural hearing loss, and chorioretinal defect. We want to look at the transcripts just to see if we want to see if all the variants are in a particular area of the gene or a particular transcript. So it's helpful to look at the number of transcripts and give yourself a sense of where all of these variants might be falling in the gene. So in this case, there's only 1 transcript. We use RefSeq. That's 28 exons. In our case, we're interested about the phenotypic spectrum for hearing loss. So in this case, just for scoring cases particularly, we want to score cases that are consistent with that type of phenotype, usually in stronger gene disease associations. But in this case, they all pose with congenital sensorineural hearing loss. And we want to ask if there's anything that's going to suggest anything about the proposed mechanism for disease. In this case, experimental evidence showed that variant protein does not form homotrimers, so potentially it could be loss of function. And this is autosomal recessive, so potentially might be more likely that loss of function could be a disease mechanism in this case. When you're evaluating the variants, as I mentioned, you want to consider the prevalence and the penetrance of the disease that you're evaluating. And then you also want to look in Nomad. In this case, it was an older curation, so the constraint scores weren't available in Nomad, so we looked in XAC. And this is pretty highly constrained for loss of function, but 2 caveats for that, and I'm sure people will talk about this. This is an autosomal recessive condition, so you have carriers. It's a little bit more accurate for autosomal dominant conditions, and also this isn't a condition that affects fitness, so it's not a super helpful metric for us in autosomal recessive conditions that don't affect fitness, but definitely good to look at in autosomal dominant conditions that do affect fitness, suggesting that this gene is constrained for loss of function. So we did a literature search for PNPT1 and hearing loss. And these are the papers that we found for variant evidence. So in 2012, There was testing done on individuals with hearing loss. They were negative for GJB2 variants. GJB2 is the most common genetic cause of hearing loss, so it's good to see that they were evaluated for that. And they performed homozygosity mapping and Sanger sequenced all genes in the linkage region. The variant here, which is a missense variant, is homozygous, is absent from both EXAC and NOMAD, so no high-frequency concerns to be worried about. The family members that were identified are homozygous for this variant. So the category that you would score it in are 2 variants, not predicted, proven null, with some evidence of gene impact in trans. Multiple family members were tested, and the phase was identified, so you can say that they were in trans. So in this case, this would be 1 point. This is the standard score. As I said, we suggest to use the standard score until you get more comfortable with the framework, and then you can modify a little bit. And so this is the pedigree here, and this is the segregation evidence. As I mentioned, they were negative for GJB2 variants. They had a region of homozygosity because it was a consanguineous family, and they Sanger sequenced all genes in this region. So I labeled here the 3 affecteds, or those in the dark colored squares that I have boxed in red, and then the 3 unaffecteds are here. You wouldn't count any of these unaffecteds up here because they don't have the same chance of receiving the variant as these unaffecteds do, so only count the SIBs in this case. They actually published a LOD score, and that was 2.7, so we don't have to calculate our own LOD score here. And this will score 1 point in the framework. So this is the little cutout of the framework here. The LOD score is 2.7, and we have 2 to 2.99 being the score here. All the genes were sequenced in the linkage region, and so in this case, we give 1 point for the segregation scoring. For experimental evidence, which was in the same paper, the wild type and the PNPT1 with the variant were localized to mouse and zebrafish inner ear tissues. So we use this for expression points here. The gene is expressed in a tissue related to the disease, and that was just a standard half point. They evaluated the function of the variant in mammalian yeast and bacterial cells, and it exhibited diminished function as an RNase P RNA importer in all the cell types and caused reduced viability in bacterial cells. And so for that, we just did functional alteration in non-patient cells, and that was a standard half point there. And so this received 2 points of genetic evidence, 1 point of experimental evidence, the 2 points being the variant and the segregation scoring. And so this corresponds to 3. It was not replicated over time, and 3 points corresponds to limited. And so this association with one paper ended up as limited, which was then finalized by the experts in the hearing loss clinical domain working group. So let's go to a moderate one now in a different disease area. So this is SOS2 and Noonan syndrome. And this was curated by the ClinGen gene curation working group. Actually, it was one of the paper genes I curated. And it was approved by the RASopathy clinical domain working group subsequently. And this is an autosomal dominant curation here. So same thing, you just want to kind of get an overview of the gene. So SOS2 is son of Cevalence Drosophila homolog 2. It has no alternative names, and it is actually very similar to SOS1. I think it's 67% identical to the SOS1 sequence, which is definitively associated with RASopathy. And this is just taken directly from the OMIM entry. You want to get a sense of the variant spectrum, so we went back to the Human Gene Mutation Database. There are 5 variants. There are 5 missense variants, and they're all supposed to be associated with Noonan syndrome. There's only one transcript that's 25 exons. A very well-established disease mechanism for Noonan syndrome and for the MAP kinase signaling pathway is gain of function. And so we would propose that the mechanism for this would be gain of function unless we see differently. Once again, consider the prevalence and the penetrance in your disease area. The probability of loss of function intolerance in this case is 1. The missense z-score is not remarkable, and that ClinGen says that of the cutoff you should be suspicious of is 3. And so at this point, it's only 1.97, so it's something to consider. However, for gain of function, you're usually looking at missense variance. And so the first paper that we evaluated for variant evidence was this paper in 2015. For testing, they did a mix of whole exome sequencing and targeted sequencing. These individuals were negative for other RASopathy genes, which is what you want to see for particularly definitive gene-disease associations. And so this is the nice table of all the cases here. The 4 variants that are boxed here in black are variants that we ended up scoring as other variant with predicted gene impact. So the standard score for those is half a point. And so with 4 of those, we came up with 2 points total. There is 1 variant here boxed in red that was de novo, and they confirmed paternity and maternity. And so that was scored more points. That was scored 2 points for a de novo variant. And they had different families in Yamamoto 2016, which is another paper. So this is a Brazilian family. They had a missense variant. The testing was whole exome sequencing. When we looked in Nomad and Exact, the variant was absent. And so we were able to score this as other variant with some evidence of gene impact, which is standard half a point. This family does not have enough segregations to score for a dominant one. You need 4 segregations in a family to score for the ClinGen framework for dominant, so we couldn't score any segregation for this family. For the next family, another Brazilian family, there's another missense variant. The testing was exome sequencing. The variant is absent. This variant is actually de novo, and so we were able to score it 2 points. And because it was de novo, we couldn't count any segregations, obviously. There was another American family that had a missense variant, also whole exome sequencing. The variant was absent from Nomad and Exact. It was a missense variant that was not de novo, so we scored this other variant type with some evidence of gene impact, which was half a point. This family actually has the same variant as the one Brazilian family. We can still score this variant if we're relatively sure that the families aren't related and that it was essentially in a founder population. You would have a difficulty if you had a founder variant, and we're still struggling as to how to score founder variants. But if you're relatively certain that this variant has potentially arisen independently, that's even more evidence for the variant's pathogenicity potentially. So you could score it if you're relatively sure that the families aren't related. And this family, as I mentioned in the previous one, does not have enough segregation, so we can't count any segregation points. So there's then a case control study also in Yamamoto 2016. For the cases, there were 50 Brazilian probens with Noonan syndrome. They were negative for known pathogenic variants in the genes associated with Noonan syndrome. And the control was exome sequencing of 107 Brazilian individuals affected by other monogenic disorders. So 5 variants in SOS2 were identified. 2 of them were in Noonan syndrome patients, and 3 were in the control population. And they did a 2-tailed Fisher's exact test, and the p-value actually wasn't significant. It was 0.21. And so we scored this 0 points because of lack of statistical significance. This could be potential contradictory evidence that you would flag and then show to experts. In this case, the experts determined that it was underpowered, and it was not substantial contradictory evidence to undermine the gene-disease association. So we just marked that it was there. We didn't score at any points, and we mentioned why the experts did not find that this is contradictory evidence. So for experimental evidence, some pretty bread and butter experiments for the MAP kinase signaling pathway were done for these variants. So they transiently transfected 3 variants into cells and looked at ERK and MEK phosphorylation by Western blot. And expression of the variants caused increase, so essentially gain of function as we had suspected. And for these, we did functional alteration non-patient cells because they're transfected into, I can't remember, HEK cells or something like that. So that was just standardized, half point. And then they did another functional assay, and so they measured GTP-bound RAS as a readout of RAS activity. And this also suggested gain of function, so stable expression of the constructs resulted in more GTP-bound RAS. And this was also functional alteration non-patient cells, 0.5 point. SOS2 null mouse was generated by targeted deletion of part of one of the domains. And they evaluated the mice, and there were no differences in compared to the control mice in lifespan, histology of 30 tissues, fertility, or development. And this could also be potential contradictory evidence, but something that we face over and over again is you need to think about the mechanism of disease in this case. So all variants were actually missense variants, and the authors suggest a gain of function, which is consistent with other genes in the disease area. And so this is not contradictory with a gain of function mechanism. This is actually just showing loss of function. And so we just scored it and mentioned that it wasn't contradictory evidence. Whereas if you had a transgenic or a gain of function mouse with no phenotype, that would be a different story. But this is not consistent with the proposed mechanism of disease in human patients. So total, with genetic evidence, there were 7 points. With experimental evidence, there was 1 point, and this totaled 8 points. It was not replicated over time, and this corresponds to the moderate section here, and this was approved as moderate by the Rhasopathy Clinical Domain Working Group. And then I'm going to end with a definitive one, and this is Clarin-1 and Usher syndrome, and it was curated by the ClinGen hearing loss group. As I mentioned, there's a decidedly different way that we curate definitive genes, and this also bleeds over into how we present them to experts. So most experts for anything moderate and below, or even strong and below, want to know every single detail of how each patient was tested, what is their ethnicity, was the variant rare in Nomad or absent from Nomad, what do computational predictors look like. But for very, very definitive gene disease associations, we literally will just put up bullet points and say how many individuals were affected and what types of variants. So the first paper was in 2001. There was another paper in 2014 that had one family with 3 affected siblings, and we'll say what we scored. And then another paper in 2011 with 1 family, 2 affected, 7 unaffecteds. There are 3 probands in this 2009 paper, and that this maxed out genetic evidence. And we will list a bunch of experimental evidence pieces, but for example, an important thing being that there is a knockout mouse that has progressive vision loss, which is consistent with Usher syndrome, which is a syndrome with hearing and vision loss. And there's a bunch of expression studies. There's another mouse model with actually a particular patient variant, and just showing that experimental evidence is maxed out in that case too. So you're really just curating until you reach definitive in this case. And so we maxed out everything and got to 18. It was replicated over time, and this was a definitive gene disease association. You could even curate this from a review if it were very comprehensive. And I just wanted to point this out. I'll stick this at the end of the workshop too. But we recently developed a very nice volunteer process for ClinGen. So if you're interested in curating genes or variants in any particular disease area, I think when we checked in yesterday, there were 30 plus groups, clinical domain working groups with experts across different areas, of some of which I listed. Feel free to take our volunteer survey and indicate that you're interested. So you can actually scan this QR code here to volunteer for ClinGen, or you can go to clinicalgenome.org/volunteer. And with that, I just want to acknowledge all the people who were involved in putting together these slides, and also some of the disease-specific groups that we worked with for the curation examples. And with that, I'll take questions, and I'll also just leave the QR code up in case anyone wants to volunteer. Feel free to come up to the mic, though, just so everyone can hear you.
So I have 2 questions.
Sure.
The first question is, could you go back to the hearing loss example? Like
Which one, Usher or the first one?
The first one. The experimental evidence part.
Yeah.
The one with mouse and zebrafish.
Oh, that's. You mean?
No, the
Oh.
one with like weak evidence.
Yeah.
Yes, over here. So my question is, at here, so I can see like the expression was determined in 2 separate organisms and was tested in 3 different types of cells. I was wondering why it was only given half a point instead of half a point each to like each of the experiment.
Mm-hmm. That is something we have discussed, and a lot of times if we see a lot of replication of the same type of experiment, we don't find that as additional valuable evidence. And so sometimes you can score it separately. Maybe if it's really, I would say, for example, if there was expression experiments in mouse and human, we would probably score 0.5 for each of those. But we found that because it was 2 animal models and it wasn't super convincing, we only scored half a point. But some groups will score like per variant or per expression experiment. In general, my rule of thumb is I like to be as cautious as possible, because if you overclassify a gene, you then start to report variants back to patients, and it could be a concern.
Thank you. My second question is about the case control, the case control. So,
Sure.
so you pointed out that there are possible biases and confounding factors, including whether there was ascertainment bias, and also whether other mutations has been ruled out. I was wondering, in these cases, how should curators deal with them specifically, and why they are confounding?
So ascertainment bias, I feel like, is just a difficulty in all disease areas that we have at this point. And hopefully with all of us and like all of these biobank studies, we'll really start to understand the penetrance of conditions. So I think you just have to be careful when you're scoring things, that oftentimes things aren't as highly penetrant as we realize. And that's another reason that non-segregations are really affected individuals that don't have the genotype. So I think you just have to keep in mind that penetrance is probably lower than we realize, because we're ascertaining all these affected individuals. And what was the other mention that you had?
The other question was like in the cases, so you mentioned that one possible confounding factor is that if other mutations in other genes has already been ruled out. Mm-hmm. I guess like in this type of study, I guess basically all studies will have already been like mutations in other well-established genes have been ruled out. So I was a little bit confused on like why this would be a confounding factor.
You know, you would actually be surprised that in some case control studies that they don't test for the most common cause of disease. I think we like to hope that almost everyone, but I've seen some pretty not well controlled or powered or unbiased case control studies. So that's just something we mentioned, that you should look to make sure that they are sequenced. And in a good study, they should be, but I've seen plenty of studies that aren't great.
I see, that makes sense. Thank you.
Mm-hmm. Any other questions? Also feel free, if it's a smaller environment or more one-on-one, to ask questions in the workshop as we go along and score some gene disease associations together.
All right, thank you, Marina.